Hi, Jeremy Cordeaux in the Court of Public Opinion. I'm just on air here to let you know that we'll be live streaming the Court of Public Opinion every Friday between 9 o'clock and 12 on jeremycordeaux.com. Please join us. We'd love to have you.
Dr. Travis Brown, what is This Medical Life all about?
This is the pursuit of knowledge as we learn about diseases from the ancient times to the present day. These are the stories of medicine.
Dr. Travis Brown, would you advise we sit down for this episode?
I would. Depends on what you're going to sit on as well. So maybe have a stool.
Oh, very good. Because it's all about the fecal biome, isn't it?
It is. So we are looking at the fecal microbiome. So this is pretty much the bacterial, I would say, breakdown, but the bacterial map, if you want to say, of what passes in our stool. And so it's a little bit hard now because what we're finding is the last few hundred years, medicine has been really good with closed questions. Right. And so the closed question is it this or is it that? And so in microbiology, we're really good at saying, okay, let's say you've got a bout of diarrhea. We look for different organisms. And now we're getting a little bit better at, sometimes we know different bacteria. Is it Shigella? Is it Salmonella? Campylobacter?
Which sound like really fancy gourmet soft drinks, by the way. But go on.
And now we know there's a whole bunch of viruses, so we also test for that. So if you've ever had that kind of a gastroenteritis and needed to have a sample, we've said, okay, well, let's send it to a PCR lab and we'll also do viruses on this. We're now getting to a stage where medicine has gotten so much information, or we're starting to gather so much of information. Microbiology is one of those areas, and genetics. It's not now, well, is it this as a closed answer? It's what do we do with all of this information? Because we're now doing the Human Genome Project, and pretty much you can spend however much money and get your whole genome mapped. And so what do you do with that amount of information? And the fecal microbiome is exactly that. We are now able to pretty much map someone's entire stool and say, well, what information can we get from that? Not just, well, even though we are doing that, what's causing the diarrhea here, or what's the problem? And this is opening up a new whole realm in medicine. And this is why we've got some experts to talk to today to help us understand this, because this was not even something that really existed when I was in medical school, and that's only not that long ago. So
yeah, it's like a dark metropolis, isn't it?
Very dark, and we're going back into ancient times for our first guest.
Dr. Piers Mitchell is co-editor of the International Journal of Osteoarchaeology and spends part of the week as a hospital consultant specialist in the NHS. He's trained in paleopathology, osteoarchaeology, parasitology, medical history, orthopaedic surgery and higher education. He was president of the British Association for Biological Anthropology and Osteoarchaeology from 2012 to 2015, and president of the Paleopathology Association, the worldwide organisation for the study of disease in the past, from 2015 to 2017. Dr. Mitchell was editor-in-chief of the book series Cambridge Texts in Human Bioarchaeology and Osteoarchaeology. He's published between 150 and 200 articles and 6 books. Along with being filmed for over 20 documentaries discussing his research on disease in past populations, and more than 1,000 news articles have been published about his work by journalists. We're adding one more to the list today because Dr. Mitchell is our guest on This Medical Life. Welcome to our podcast.
Lovely to talk to you.
May we start? Can I get you to tell us about your studies you've conducted on analysing our ancestors' microbiome?
Well, the best way to study an intestinal microbiome if you're looking at ancient samples is to make sure you've got some ancient feces that's human. So we can do that by looking at pieces of ancient feces that have survived in archaeological settings, such as coprolites. They may be dried, or they may be waterlogged, or they may be mineralized. That helps them stay in a distinct piece. And another way we can do it is to look at latrines. So you have the mixed up feces of many different people that used that toilet in the past, and that will be mixed together and become sediment that you can find underneath a latrine seat, for example. And those are the two main ways that we can look at the microbiome from the intestines from the past. So one way of doing that is to look down the microscope and see the remains of the organisms that were there. And that works for larger organisms, particularly intestinal parasites. So the eggs of intestinal parasitic worms like tapeworms and so on, they often are quite tough. They're large enough to be visible with digital light microscopy, and we can find those kind of organisms in those samples. However, many organisms are far too small to see on microscopy. And so for that, you really need to study the ancient DNA, so the fragments of their genetic material that will survive in those samples.
Can I ask then, it boggles the mind that these specimens have survived this long. So which cities were these conducted in?
Well, things survive very differently in different environments. So some of the remains that have been studied are pieces of coprolite that were deposited in the back of caves. And so they are very dry environments. You don't tend to get tree roots and plants breaking the things down in the soil as you might do outside a cave. And so long as there isn't water flowing through the cave, that can result in those coprolites surviving very well. However, in other settings, waterlogging can make the fecal material survive very well because it makes it anaerobic, takes away the oxygen, makes it much harder for insects and other organisms in the soil that might break these things down and eat them as part of their lunch. So waterlogged coprolite or waterlogged latrine sediment helps things to survive very well. So you're quite right that ancient DNA doesn't survive as well as modern DNA. So if someone were to take a swab of DNA from your cheek or a blood test or something, you would have entire whole structures of your genome. However, ancient DNA, all the fragments are broken up, and so you tend to find 50, 100, 150 base pair fragments of DNA. And that's why to study those, you really have to have a very clever computer that can stitch them all together and recognize that this bit is the same as that bit and stitch them together and make a computer-generated whole genome just by the large number of overlapping sequences that match.
Can I ask then, you've looked at ancient microbiome and modern microbiome. Can you tell us what actually is the difference between the two, if there is any?
Well, part of the differences in microbiome is how you live your life and what you eat. And so there are studies comparing hunter-gatherer populations in the world today with modern industrialized populations. And they've looked at the differences that those people have. And then when we look at ancient microbiomes, we can see what we find in those ancient samples, and we can see how similar they are to hunter-gatherer groups today and industrialized populations today. So the majority of the organisms that we find in our intestinal microbiome will be made up of bacteria, archaea, which are like very simple sort of ancient bacteria. There'll be some fungi, there'll be parasites, there'll be viruses and so on. And some of those, the genetic material is easier to study than others because it's preserved better. So quite a lot of the microbiome work has been based on the bacteria, the archaea, and the parasites. The work comparing modern hunter-gatherer microbiomes with modern industrialized microbiomes tends to indicate that the hunter-gatherer microbiomes have a very broad and diverse microbiome, lots of different organisms in there. They also tend to have parasites present. They also tend to have bacteria that are quite good at breaking down fibrous materials. If you compare that with modern industrialized microbiomes, we tend to find less diverse range of organisms. We tend to find more organisms that have genes that are give resistance against antibiotics. We tend to find few or no parasites, and we tend to find bacteria that are much better at breaking down mucus than breaking down fiber. So it would suggest that the differences in diet that people have, whether you're in an industrialized society or in a hunter-gatherer society, is going to reflect what organisms are successful. Now, if we compare that with work on ancient microbiomes, there are obviously much less work done on ancient microbiomes than there is in modern microbiomes, because it's not that easy to get ancient samples. And it's more of a challenge to actually get the DNA out, because it's not all complete like the modern work is. The study we did with some colleagues from Germany, we looked at not only the parasites that were present, but also the ancient DNA of the microbiome of 14th and 15th century latrines, one from Jerusalem, so a very ancient city in the Middle East, and then one from Riga, which is capital of Latvia today. And that latrine has only been in a city that was only founded about 200 years before. So it was a very young, fresh, sort of vibrant population, as opposed to a population that had been there for thousands of years. And what we found was that the microbiome from both those medieval populations shared characteristics of industrial and hunter-gatherer societies today. So in other words, it was a bit like an intermediate form. So in the past, they were starting to move away from the pure hunter-gatherer microbiome, because they were living in cities, and especially in Jerusalem, they've lived there for thousands of years. But it hadn't yet reached what we find in modern industrialized populations. There was another interesting study that was done looking at coprolites from caves in the southern parts of the United States and Mexico. And they dated from about 1,000 to 2,000 years ago. And what they found there, although it's a completely different continent to where we were studying, and they're doing coprolites as opposed to we were studying latrines, which gives you a sort of mixed set of the microbiome for the whole population. What they found in those coprolites was that there was, again, more fiber degrading capacity in the organisms, less mucus degrading capacity in the organisms, and also much less antibiotic resistant genes in those organisms. So again, shows how the diet they were using affects what they were eating, and the fact that they weren't using antibiotics in the past in the way we do now, although they could have, of course, still been eating leaves of plants that may have had antibiotic properties. We see a lot less disease resistance genes in the ancient microbiomes than we see in the modern ones.
Now, one of the things that keeps on coming up when I'm reading these articles is parasites. And it's sort of hard to think, because we think parasites are just terrible and that they need to be gotten rid of. Is that almost your interpretation when you look at the ancient microbiome? Do you think there's a difference because we now don't have parasites?
Yeah, so it's absolutely crazy that we don't have parasites. It's because you can see them, and people don't like wriggly things.
Yes, exactly.
Whereas you can't see the bacteria, you can't see the archaea, you can't see the viruses, you can't see the fungi in your feces. So people are okay with that. So you can buy little pots of yogurt that are marketed as having healthy bacteria in there. But you don't find little pots of yogurt being marketed as having healthy worms in there, because people don't like that kind of concept. But they're an intrinsic part of our evolutionary microbiome. So if you look at how humans evolved from other ancestral primates in East Africa, and you look at the kind of parasites those modern primates have, and you compare that with the human microbiome and so on, we should be expecting to find parasites in a normal human microbiome in the way that you find them in gorillas, and you find them in chimpanzees, and you find them in baboons. And so the fact that we've tried to remove those parasites means that there's a big chunk of our microbiome missing. Now, some of those parasites might do you harm. So I can understand the logic in not having those. A lot of those parasites don't do you any harm, especially if you have them in low numbers and you're well fed. So absolutely, if you're a little child in a part of the world where they're not getting enough food and they have lots of diarrhea and other diseases that might make them ill, then having a load of parasites can definitely be bad for you. But if you're a well-fed person in a modern society and you have a few whipworm in your intestines that cause you no symptoms, you just share a little bit of your food with them and they don't cause any problems, then that's probably something that would be better for us rather than clearing our intestines of every worm just because people don't like the idea of them being there and don't like seeing them in our feces occasionally, when it's something we've evolved to have from an evolutionary point of view.
Can I just clarify? We've gone from the scale of them being negative in that first example and then being neutral in the latter example. Is there something on the positive edge of the ledger that they bring, or is it just we can mutually coexist and that's good enough?
Well, from the point of view of diseases, I hope your colleague that will be talking about the clinical impact of microbiomes today, they may well talk about how people have even tried to reintroduce intestinal worms to treat certain diseases. So some worms have minimal inflammatory impact on the body, especially the ones that stay in our intestines and don't migrate through the body, which some do. And so if they don't cause symptoms, then you could argue that them being part of your intestinal microbiome might help to calm down the other bits of your microbiome that may overgrow and cause mischief when you don't have a good balance. And one of the problems we have is that cause and effect is not always easy to tell with disease, but we certainly know that different microbiomes have been noted in people with different intestinal diseases. So people with colorectal cancer, people with inflammatory bowel disease, people with irritable bowel syndrome, people with obesity. So all these different groups have been noted to have slightly different microbiomes. And what's challenging is teasing out which ones of those are causative. So if your microbiome might make you more prone to be overweight or more prone to have bowel cancer, or a result of the disease process that might make some organisms flourish more than others. But there does seem to be that people with different intestinal diseases do have different microbiomes or are more likely to do so. And so consequently, the idea of manipulating your microbiome to reduce your risk of a disease happening or to help reduce symptoms or help those diseases to settle down by rebalancing the microbiome to something we're used to at an evolutionary level seems like a very sensible idea, so long as you can avoid causing disease from introducing things that you don't currently have.
There's also a theory that with the reduction of parasites in our gut and exposure to it, that there is a hypersensitivity has been on the increase. And so sometimes some people say because that causes an eosinophilia and IgE response in our body, that's now tipped over to because we have reduced parasites, we actually have much higher levels of hypersensitivity. And so this is sort of an interesting area. I'm not quite sure. It's quite hard to say because I don't think right now we've sold you, Steve, on going and getting a parasite at the moment.
Actually, you know what? Piers has, and I do marketing as my day job. I think parasites suffer from a marketing problem. We need to rename the better ones.
Okay, sounds good. Can I ask then, was there any unexpected findings in your analysis, Piers, with these?
Well, there are a lot of challenges with looking at ancient microbiomes. And one of them is that a large chunk of what you find, you have no idea what it is. So in the study we did on our medieval latrines, about half of the DNA we found, we did not know what it was. And about a quarter of it looked like being soil environmental organisms. And about a quarter of it was probably the actual microbiome of the people that went to the toilet in it. So we do have this challenge of what is that half that's missing? Are we discovering new organisms that don't exist anymore? Or is that environmental organisms that people just haven't studied before? So they're not in the computer databases for the computer to tell us, oh, this is X or Y bacteria. And because one of the issues is there's lots of money for studying. If there's an outbreak of dysentery, there's lots of money for going and studying the genome of that pathogen. But there's much less money for studying bits of soil around the planet in different places to say what's the normal organisms that you find in the ground in this part of the world. Because that's less sexy. But it's very important if you're trying to understand what organisms are going to be affecting people, what might be pathogenic, what might be good for you, and how microbiomes might vary in different parts of the world. Because we all know that kids sit there and eat soil and they pick up things off the ground and everything else, and so a lot of their microbiome is going to be affected by the local stuff in the soil. But that kind of thing is a challenge. How do you differentiate pathogenic organisms or commensal microbiome organisms from this half that we don't know what it is? And if you study coprolites from caves, for example, you're probably going to get fewer microbiological input from the environment compared with if you study stuff that's been sitting in the soil for the last 2,000 years, where all the creepy crawlies are going to move in, and try and break things down. But the key points are sort of teasing out those. So the things I've found fascinating so far have been the rise of the microbiological resistance genes, the change in the balance of the different organisms that we're finding, way that you find if you go back in time, you start getting these sort of hybrid microbiomes that have some traits of hunter-gatherers and some traits of the industrialized microbiome. And the fact that we found differences between an ancient city like Jerusalem and a modern town like Riga, as in 200 years after it was formed, we're finding different sets of microbiomes in those different populations. But whether that reflects the length of time people have been living there, or whether it reflects that one in the Middle East and one's in Northern Europe has a colder, wetter climate versus a Mediterranean climate, all those kind of things, it's a little bit hard to tell what the cause is. But finding those differences in the microbiomes of ancient populations is something that I find really fascinating.
Indeed it is, and I think this has set us up beautifully for the rest of this episode. So, Dr. Piers Mitchell, I'd like to thank you for two reasons. One, the fascinating insights and that historical aspect to this, and two, for me to know tomorrow morning when I sit on the latrine that I will be contributing to the scientific discoveries of your descendants in a few hundred years from now.
I'm sure they'll be very grateful to you for that.
Thank you, Dr. Piers Mitchell.
Bye-bye.
Our next guest is Dr. Vincent Ho. He's a gastroenterologist who practices at Campbelltown, New South Wales. He has a PhD in biological markers of colorectal cancer. He's an associate professor at the Western Sydney University and is the head of a research laboratory. In his practice, Dr. Ho sees patients associated with irritable bowel syndrome, celiac disease, and inflammatory bowel disease. Dr. Ho is also on YouTube as the Gut Doctor, written a book called The Healthy Baby Gut Guide, and has an education hub on his website, gutdoctor.com, which is GUTDR.com. Dr. Ho is a returning guest today on This Medical Life. Welcome back.
Thank you so much. Thank you for having me back on board.
I'm looking forward to it, and I'll sheepishly listen in because I did have a look at your Gut Doctor site, given that I've started taking a general probiotic daily, and I've realised I've probably got into that like a true amateur, and I should have chatted with my GP first.
So can I ask then, we're looking at the fecal microbiome, and can you tell us what is the fecal microbiome?
It's a great question, and I think we should start off by defining what the microbiome is and the closely related term microbiota. So these two terms are often used interchangeably, but they do have different meanings. So put very simply, microbiota is the actual bugs or microbes themselves, and microbiome is the microbes and their genes. So that's a really important point. Now, the gut microbiome itself is made up of more than 100 trillion different microorganisms. Most of that's bacteria, but also fungi, viruses, protozoa. And the fecal microbiome is really a surrogate measure of what's occurring in the gut. So often when we talk about the gut microbiome, we're actually in practice looking at the fecal microbiome because it's usually fecal samples that we are examining to help determine the composition of our gut microbes.
So there is something called the Human Microbiome Project. Can you tell us a bit about that?
Yeah, this is actually an interesting project. And in many ways, this was the natural successor to a very well-known project called the Human Genome Project. And this was an international race to map out the human genome. And you may recall that was a big race, started in the early 90s and finished around 2003. And it was around that time that it finished, that due to the improvements in DNA sequencing, that scientists started examining the human microbiome. And the advantage of examining it with a genomic-based technique is it allows the microbiomes to be examined without culturing any of the microbes. And in fact, some of the microbes are not able to be cultured readily. So it had that real advantage. And so it was around 2007 that the National Institutes of Health in the United States initiated this human microbiome project. And this project went for about 10 years, went from 2007 to 2016. And it has been instrumental in providing information about the human microbiome. Just in brief, there were two phases to this big research project. Phase one was in determining the microorganisms that make up the microbiome. So that work involved surveying the microbiomes of 5 different regions, so skin and mouth, the gut, of course, the urogenital tract, and the nose. And they did this for healthy individuals, and they found that the microbiome composition of each body site, not surprisingly, is quite distinct. So that was phase one. And then there was phase two, which was working out what the microbial communities, what they were doing in the body, how they were interacting with the human host. So that phase two involved analyzing the functions of the microbes, and they examined it in particular with a few disease conditions. So one was preterm births, another was inflammatory bowel disease, and the other was the onset of type 2 diabetes. And what they found in brief was that certain microbes are linked to these disease conditions.
So then we're looking at pretty much previously we've talked about our ancestors' guts, and now we're looking about our microbiome. Is there been any surprising findings about this that were unexpected, or was this all as the scientists believe was going to happen with this?
Yeah, so there are some surprising aspects from the human microbiome project. And I think one of the surprising findings was that microbes contribute more genes responsible for human survival than our own human genes. So microbes have more genes, and that was amazing. And it's estimated that bacterial protein coding genes, they're thought to be about 360 times more abundant than human genes. So it's incredible, really. The other interesting and somewhat surprising aspect of the research, they found that the metabolic activities of microbes, for example, things like digesting fats, they weren't always carried out by the same bacterial species. And it's actually the presence of the activities that seem to matter more for the microbes. So it's not necessarily a particular type, but it's more about microbes themselves having a cluster of activities. And I think another point about the research that was gleaned from the Human Microbiome Project was that there seems to be a natural equilibrium for the human microbiome. In other words, often you get disturbances because of diseases and antibiotics, but what is found is that the microbiome tends to go back to a state of equilibrium, even if the composition of the microbes have changed. There's a natural tendency to go back towards equilibrium.
Mm. Now, looking at, you've already mentioned a few diseases, but can you go through the diseases that have been linked to gut flora?
Yeah. So there are quite a number of human diseases that are associated with altered gastrointestinal microbiota. So things like obesity, diabetes, cardiovascular diseases, cancer, hypertension, allergy, autoimmune diseases, and inflammatory bowel disease. There's this state called dysbiosis, and that basically means variation in gut microbiota composition. That's been described in many different diseases. And so we can often see this state of dysbiosis in many of the different conditions. But increasingly, we're seeing in many neurological disorders, conditions like Alzheimer's, autism spectrum disorder, multiple sclerosis, Parkinson's disease, and even stroke, that the microbiome, say, best way to describe a susceptibility factor, that there are linkages to these conditions that we would not normally think of as linked to the microbiome.
Mm. So does this project have an end in sight? Is it, now you mentioned 2016, is it still sort of continuing on, or is it branched into a whole other range of research areas, or is it stopped?
Well, the answer to that is yes and no, because we know that the Human Microbiome Project officially ended in 2016, but the work from that project is still continuing. And there is just so much about the gut microbiome that we are yet to discover. And in fact, I would say that we're at the beginning of this fantastic voyage.
Can you tell us now, for the practicing general practitioner today, or for other clinicians, is there any applications from these findings from this project that they could implement today that from this project?
Yes. So there are a few key points that I believe that the clinicians should be aware of. So we know that what characterizes a healthy microbiome is now fairly well understood. And there are three key features that characterize a healthy gut microbiome. The first is diversity. So that refers to how many different species of bacteria there are. So a healthy gut microbiome should be diverse. And we know that one of the best ways to increase your microbial diversity is the richness of your diet. So this is a practical advice here that if you have a rich and diverse diet, that is linked to a diverse gut microbiome. Secondly, know that a healthy gut microbiome should be stable and resistant to changes such as bad diets. So the stability of a gut microbiome is a reflection of gut health. It should also be resilient, and that means it returns to a healthy state after a disruption. So, for example, if you've had antibiotic use, then of course there can be disruption of the gut microbiota, but there should be a natural resilience for it to return to a healthy state. And on that note, there are strategies that clinicians should be looking at when it comes to the gut microbiome. So I use the term modulation, which refers to making changes in the microbiome. And some of these changes, for example, include the use of probiotics. So that's live microbes that have beneficial qualities when ingested. Prebiotics, which are compounds, food that are the fertilizer, good bacteria in the gut. And postbiotics, which is really new, but that refers to the metabolites, the waste products of healthy microbes. So an understanding of these biotics is quite sensible, because these concepts will be talked about again and again and brought up to clinicians by their patients. I think it's important to be aware of the fact that the gut microbiome can be altered in other ways. Selective antibiotics can be used to treat dysbiosis, that imbalance in the gut. And there are new emerging technologies. For example, fecal microbial transplantation, which is the transplant of gut bacteria from a healthy donor to the gut of a person with a compromised microbiome.
Just reflecting on that advice then about the diversity of diet as a good way to stimulate a healthy gut microbiome, when I do talk to my GP about my probiotics that I've started on, I might ask them for some recipes and a meal plan. That seems to be something that would be part of the mix. And
yes,
but just that shopping list of diseases linked to the microbiome that you've talked about, is that what the intrigue about that, is that what led to your interest in this area? What's made Vincent Ho so attracted to this field?
Yeah, so a key area of mine, I guess a key area of mine is in microbiome modulation. And one research area of mine that I'm quite passionate about is actually fecal microbial transplantation. Because that is one intervention that is able to result in a massive diversity boost. So if we're looking at changes in the microbiome, one of the most effective ways to alter diversity is by a fecal transplant to boost diversity. And so because there's been a lot of research in fecal microbial transplantation, or FMT, we use the FMT as the abbreviation. This led me to think, well, we know that it works for certain conditions. Clostridium-difficile, it's very well known for that particular condition. What other conditions could be used used in. And so I became very intrigued about its potential for irritable bowel syndrome, particularly IBS that's refractory to common treatments, such as the low FODMAPs diet. And then I also became intrigued by effects of FMT on cognition. And so at Campbelltown, we are carrying out, through Western Sydney University, we are carrying out a single center, randomized, double-blinded control trial looking at FMT for IBS, that's failed conventional therapy. And the sub-study of this is examining FMT in IBS patients that also have mild cognitive impairment. And the reason why this is interesting and so intriguing is there's been case reports that FMT has improved cognition in Alzheimer's disease, and presumably through microbiome modulation. And we're interested in studying in a randomized, double-blinded, placebo-controlled trial whether or not FMT can actually improve cognition in those that have mild cognitive impairment. And we're using validated questionnaires such as mini mental state examination and the Montreal Cognitive Assessment for that purpose.
Vincent Ho, this is a psychological overload. It's so linked to so many things. Let's just take a short breather, and come back in a moment and go just a little bit deeper. We're continuing our discussion with Dr. Vincent Ho now regarding the fecal microbiome. I want to come back. You've mentioned probiotics a couple of times, and I do see a lot of marketing, which has been effective for me. Does it actually change or have any impact on someone's gut flora? Because you did use air quotes earlier when you were talking about the effect they can have.
Really great question. So the simple answer to that is that all the evidence support probiotics as not being all that helpful for someone who's healthy. Healthy, there's really no need for probiotics. However, we know that probiotics have efficacy in a number of conditions. For example, antibiotic-induced diarrhea, there's clear evidence that the use of probiotics can shorten the duration of symptoms. One key point about probiotics is that it has to be administered in adequate amounts, allowing for enough bacteria to arrive at the colon to exert their health effects. And this is the reason why you will see probiotic capsules often contain many billions of coliform units. And the concentration of bacteria, they exceed what is normally found in fermented foods. So often in fermented foods like yogurts and miso, people will say, oh, this is a probiotic. But the commercial probiotics have a very high concentration, and it's thought that concentration is needed to enable the bacteria to survive stomach acid and to colonize the gut. So that's the first point to say. The second point to say is that if it does colonize the colon, then yes, it is efficacious and it actually results in true colonization. A good example of this, breast milk. So we know that breast milk itself has a number of natural probiotics. Often these are bifidobacterium or lactobacillus strains, but they've been shown for multiple health benefits for the infants, such as reducing allergy risk, particularly in those infants that have pre-existing allergies or if they've got a parent that has an allergy history.
So coming back to the point of that you left off with last time, which was C. difficile infections. So this is antibiotic-induced diarrhea. And you mentioned the fecal microbiome transplant, FMT. And so do you think that this is going to be a common clinical application of treatment in this infection?
The answer is yes. In fact, FMT is already happening. It's used in several hospitals around Australia for the treatment of recurrent C. diff infections. So clostridium-difficile infection, or C. diff as we use it as an abbreviation, is a type of infection that can lead to a very severe inflammation of the colon, or colitis. Now, we know that FMT has a cure rate of around 80 to 90% for recurrent C. diff infection, which means that the vast majority of patients will be cured of recurrent C. diff with just a single treatment. There are some patients that may need more than one FMT treatment, but the vast majority can be cured with just one single treatment. And that's really amazing.
Can you actually tell us what's involved? Because I'm sure the mind reels a little bit about where the sample comes from. Is it from the lab? Is it from a donor? Yeah, I'm sure Steve has a few ideas.
I'll just step outside for a couple of minutes.
But can you tell us what actually is the process and how is the sample or the donor acquired? Can you tell us about that, please?
This is a terrific question. So in sourcing out a donor, normally the donors are related to the recipient. So, for example, it could be a partner that's contributing their donor stool. It might be another family member. So often it's someone who's related. If they're not related, it may be a healthy volunteer that's contributing their stool. What I will say is that there's a very extensive process of screening. There's a whole battery of blood and stool tests that are required to rule out infections. And there are some questionnaires that are administered to screen out potential diseases in donors. So we need to make sure that the donors, whether they're related or not to the recipient, is a healthy person that is unlikely to pose a significant risk of passing on any infection. And that's a really important point. As far as the methods of delivery is concerned, well, it obviously makes sense that we should be giving it to patients in a palatable form. So FMT has been given and was given thousands of years ago to people. And in fact, it was in China where FMT was first reported. They given it in a soup form. But I think that's not going to be very attractive. No. And so these days, it's usually given by either an enema at the time of a colonoscopy. So that can be delivered right up into the ascending colon. It can also be delivered by the upper gastrointestinal route, not orally, typically through the use of a nasojejunal tube. So we can put a tube down through the nose, down to the small bowel, and administer that way. Another way in which it has been administered is through the use of capsules. Yeah. Some people will call them crapsules because it contains fecal matter inside.
Yes.
Probably the most common method in Australia is via colonoscopy, and there's a good degree of evidence in literature that supports that delivery method. What I will say is that in the preparation process for FMT, patients typically, especially if you've got C. diff, will take an antibiotic such as vancomycin for a few days, and they will stop that antibiotic about 24 to 36 hours before the procedure. Now, before a colonoscopy, patients will have bowel preparation before the procedure, of course, and on the day, it's very common for the patient to be given a medication such as loperamide, which helps to retain the stool. So loperamide is a drug which will slow down bowel movements, and so it tends to retain the stool in the colon. And that's essentially the process of FMT.
Mm. Can I ask then, when we're looking at the actual diseases that you're wanting to manage with someone's gut flora, is there a therapeutic goal of actually saying we need to change your gut flora and then test it from A and then do a test B and see if you're able to change it to manage the condition?
Yeah, so when it comes to clostridium-difficile infection, we know from the research that's already been performed in that condition that there is a dramatic change in the microbiome of the recipient or the patient. So we know that there is a great burst in diversity. So when it comes to clinical routine use, we don't need to do a microbiome analysis after FMT in that condition, in Clostridium difficile, to confirm that there's been a change. What we're really looking at is an improvement in symptoms. That's the most important thing. If someone with colitis, if they're exhibiting an improvement in their symptoms, reduced episodes of diarrhea, a reduction in bloody stool, all of that is relevant. And it's far more important that we assess for a clinical response in those patients than for microbiome changes, which we know are going to happen. The increase in diversity, that burst in diversity, is going to happen after an FMT. What we're really looking at is clinical improvement.
Then coming back to C. diff, then when, because clearly this is quite a common problem, When is someone considered appropriate for FMT therapy with a C. diff infection?
So the indication for FMT C. diff infection is for recurrent C. diff infection. So where they've failed antibiotic therapy, it's ironic because clostridium-difficile infection is it predisposed by antibiotic use. At the same time, first-line treatment is the use of antibiotics such as metronidazole or vancomycin. So currently, we use antibiotics such as metronidazole or vancomycin as first-line treatment for C. diff. There are some other antibiotics that can be used if they fail, such as fidaxomicin, but there are going to be a good proportion, and it's thought that about 20% of people that had treatment for C. diff end up failing. And for that proportion of the people that fail conventional C. diff treatment, then FMT is indicated.
Now, when is it appropriate for someone to request a test of someone's fecal microbiome?
That is an excellent question because at the moment, although the test is used by clinicians, the fecal microbiome analysis is still considered experimental. And there are reasons for this. We are very good at generating a lot of data from genomic sequencing. And this data is known as bioinformatics data. It's very dense and it's quite difficult to analyze that without significant computational resources. So it's difficult to determine just how change in one part of the microbiome might affect another part and just how all that's related to disease conditions. However, I like to think that artificial intelligence has a tremendous role to play because I think that we will get to a point in the not too distant future where AI will be able to analyze a whole pipeline of bioinformatics data in a very efficient manner that provide meaningful clinical information. So we're not ready just now for it, but I think we will be in the not too distant future.
Now, this feels like an area that's primed for pseudoscience, to be honest. It feels like an area that's ready for the internet because then you'll get a whole bunch of influencers and people come on and say, change your microbiome, here's the new product and everything, here's the new powder I have, here's the new drink we have. Is there any reputable sources for doctors in this area who's wanting to keep abreast of all the information?
Yes, there is. And one of the best sites I found is actually from the NIH itself. So it's from the NIH National Human Genome Research Institute. And there's a web page which has actually got some really good tools for learning and teaching the microbiome.
We'll put that in the show notes. Now, just wrapping it all up, is there any final thoughts or last ideas you have this area for GPs to have there as their take-home message from this episode?
Yes. So awareness of the human gut microbiome is becoming increasingly mainstream. And I think that GPs are going to be approached by patients, if they haven't already, about their gut microbiome and how it might link to their symptoms. And I would encourage GPs to become open-minded and help to educate their patients. And as you've just pointed out, this is an area which there are already experts in pseudoscience and alternative medicine, along with slick marketers, social media influencers. They can paint the gut microbiome as this universal panacea for a whole range of different ailments. But that's not the case. And we need to make sure that our patients are aware that they shouldn't be abandoning, for example, cancer treatments for a blend of natural probiotics sold by a slick pseudoscience marketer. So an awareness of the gut microbiome is really important, especially because patients themselves are now aware. It's becoming very popular, very mainstream. And I think the more awareness that you have, the more knowledge, the more useful it will be for your patients.
Excellent. On that note, I really hope, what a fantastic episode. I hope many GPs, specialists and medical students have a chance to pull up a stall and listen in to this particular one. Dr. Vincent Ho, thanks again for joining This Medical Life.
Thanks very much.
This Medical Life is recorded in the Talked About Marketing Studios in Adelaide. For show notes and more information about the podcast, visit thismedicallife.com.au. You can contact the hosts via Twitter. Dr. Travis Brown is at Dr. Travis Brown. That's DR for doctor. And Steve Davis is at Steve Davis. Editing and production is by Tim Whiffen. Design is by Tom Buzenjut. This has been a Pathnotes Proprietary Limited production.
Hi, Jeremy Cordeaux in the Court of Public Opinion. I'm just on air here to let you know that we'll be live streaming the Court of Public Opinion every Friday between 9 o'clock and 12 on jeremycordeaux.com. Please join us. We'd love to have you.